All right. Good afternoon, everybody. Today I'm going to give you, going to scale back a little bit. Tom gave a great overview on some of the theory and statistical background knowledge on the different meta-analysis methods. And I am going to then give you a nice practical applications of how you can implement the activation likelihood estimation method in the BrainMap environment. Okay. So just to refresh, what we were doing is that we're dealing with coordinate-based meta-analysis methods. You've seen the papers where we publish all these lovely pictures of brains, and what we get from that are the coordinates of activation. These are their X, Y, Z locations in standard space. And so BrainMap is a resource for anyone interested in meta-analysis to go ahead and take advantage of. BrainMap is an archive of these peak coordinates and the metadata that are in the literature. If you're interested in a particular question, say, for example, what brain regions are typically activated in a memory task in schizophrenia patients, then BrainMap is a resource that potentially can help you address that question. And so our website is available right here. Okay. We have a quick author search that you can take advantage of in the top right corner, and then below that it gives a representation really quickly. BrainMap is actually two different databases, a functional imaging database and a structural imaging database, and the statistics here for the functional database are given. And then below that we have links to 3 different software applications. The first one is Scribe. So if you want to do a meta-analysis, you're going to first go to BrainMap and see what papers are available on that particular topic. But you'll see that some papers are in the database already, some may not be. Scribe is the tool that you would use to submit additional papers that you find, as Felix described, using PubMed or any of the other citation indexes, to put additional papers into the database. Sleuth is the second application that allows you to then directly search the database to find studies that you're interested in. It can search the functional database or the structural database, but those are independent searches. And then Ginger Ale is a tool that allows you to actually perform the meta-analyses using the activation likelihood estimation method. So, to give you a brief overview of Scribe, you can go to the website, brainmap.org/scribe. You've got the downloadable application up in the upper right-hand corner. Then you have a list of resources over on the right, some sample files, the manual to help you use the software, and then, yep, there's the manual. And then there's also some links to definitions of the taxonomy so that you can understand some of the terminologies that we're adhering to in the BrainMap environment. So I've got a brief little video here for you to see. I'm clicking on a Scribe file right now and opening up the application because I want to give you a sense of one of the advantages of using BrainMap is that we have a really robust taxonomy, a way of representing the metadata associated with the given study. Scribe is the application that gives you a bird's eye view into that metadata. We have information on our subjects, including sample size, gender, handedness. We have information on our conditions, stimulus response instructions. We have information on yes, external variables, additional variables that are collected. Basically, all of the metadata that is extracted by hand from the paper to give you a sense of what was done in a particular study, all of that is coded using the Scribe application. This also includes brain template, and then you pop into the experiments panel, and that's where you actually give information on specific contrasts. So if you're comparing, if it's an n-back task and you're comparing a 3-back versus 1-back, all of that information goes into the experiments panel. You've already set up your subjects information and your conditions information. This is where you click things over so that everything makes sense. One of our high-profile metadata fields is paradigm class. That archives the task. Next is the behavioral domain, which archives the cognitive construct that's isolated by the contrast. So the sense from this video is that there is a wealth of metadata coded for every single paper. This is one reason why it takes us a little while to get studies into the BrainMap database, because we have such a wealth of metadata associated with each study. And this is tediously taking you through all of the different behavioral domains. And then the last page here are the coordinates themselves. So you see we have all this stuff that at the end, then go the coordinates, which are the things that we're using to actually meta-analyze the data. So that is a brief overview of Scribe. As I mentioned, there are some resources on Scribe's website that give you some definitions and some examples of both the behavioral domain and the paradigm class. There's also a new website, brainmap.org/taxonomy, which goes into many more features of the metadata. Next up is the Sleuth application. Again, you go to brainmap.org/sleuth. You've got the downloadable applications on the right, some information about getting a user ID and password so that you can log into the database, and then the user's manual, and then a quick little demo here of using the Sleuth application. So go ahead and click the Allow button. What we did when we were designing Sleuth is this was back in the early release of iTunes, and it was really cool to construct your own searches. So we mocked this up similar to the iTunes interface. You can construct your searches as either and or any combination. And then you download papers, check to make sure those were the right search criteria that you wanted, then download them into the workspace. And so you see we move through the different tabs up on top. Once you're in the workspace, you can then access the full metadata information for each of the studies that were returned based on the search criteria that you input. And so you see here you can flip through all those different fields on the right. The final panel is the visualization in a glass brain of the different coordinates. Then there's some extra toolbars up top that allow you to export your foci in text format. This is a key feature if you are doing a ginger ale meta-analysis. You use Sleuth to search for papers that you're interested, filter the results accordingly, and then you actually export them in a text format and do the meta-analysis in a separate application, ginger ale. So this is what the text format looks like that's exported from Sleuth. This is specially formatted so that we are making sure that you have a record of the paper that you exported the foci from, the contrast, as well as the number of subjects. Ginger ale uses all of this information to run its various parameterizations, and so all of that is shown here. Okay, so now we want to get to the ginger ale application to give you an idea of how it's used. And so if you're searching the BrainMap database, and let's give an example of what ginger ale is actually doing. Tom covered this. I'm going to kind of go over it graphically to give you a different sort of representation. Say your search criteria were for a paradigm class of face monitor/discrimination, so you're interested in face discrimination studies. And if you're interested in control populations, you don't want to search subjects controls, because that's going to give you all of the search results in BrainMap where a control population was used, which that's for all healthy studies, but it's also for all patient studies where you're contrasting with healthy controls. So what you want to use is the field context equals normal mapping. That's a good way to filter your search results so that you only get healthy individuals. So you use Sleuth, and it's going to find you a number of different papers. This is just an example of two different contrasts from one particular paper: memory greater than gender, gender greater than memory, from Ken Paller's study back in 2003. The important point, and Felix addressed this, is that you can do the search in BrainMap, but you're going to need to use Sleuth or some other means to filter your meta-analyses so that you're pooling contrast that makes sense to include in a meta-analysis. So after searching and then filtering the results, you end up with 396 experiments of healthy controls that participated in a face discrimination task. And so these can be downloaded in the Sleuth application. You can tell there's a lot of coordinates associated with these 396 different contrasts. So we want to do a meta-analysis using the ALE algorithm in ginger ale. And as Tom addressed, we're looking at where these foci converge in the brain. And so the ALE algorithm models each coordinate location, each XYZ, as a 3D Gaussian distribution. And the ALE citations are over listed on the left. So graphically what this means, you can take your coordinates, look at them, model them with a Gaussian distribution, compute your ALE scores, and then determine statistical significance and threshold the final ALE images. So that's what the ginger ale application actually does. If you want to utilize it, you go to brainmap.org/ALE. Again, similar to the other applications, we have the downloadable applications up in the right-hand corner, and then we have the user's manual. And because we are doing ALE meta-analyses, it's important that when you take your final output results from Ginger Ale, you overlay them onto a standard brain space. You can do the meta-analyses in either MNI or TaylorX space, and we have standard templates for you to download on the lower right-hand corner of the page. Let's walk through the application itself. Double-click on Ginger Ale, pulls up the application, and open Foci. Select the file that you output from your sleuth searching and filtering, load that into the Ginger Ale, select various different parameterizations of statistical significance. We have a number of different ways of thresholding the ALE images, and then click Submit, or Compute, whatever that said, and let it run. So we've tried to make this as user-friendly as possible so that you spend the majority of your time doing the literature search and filtering all the different contrasts, because the most important point, again, that Felix addressed, is garbage in, garbage out. You need to spend the time thinking about what is your meta-analysis question actually trying to ask, and are the contrasts that you're putting into it actually asking that question. So in terms of the actual computation, we'd like to make it as user-friendly as possible because you've already done an enormous amount of work before you get to the ALE application. So ALE outputs a number of different files. It outputs the ALE values themselves that are unthresholded. It outputs a text summary of the thresholded images, the thresholded images themselves, an image of the p-values, and all of these different files that are output are well described in the manual and on the BrainMap user forum so that you know what the data are actually being produced, what each of the files are. Once you have those files, you need to view them somehow. You can use your favorite image viewer. Our BrainMap supports the development also of the Mango application developed Jack Lancaster, and it provides a nice and easy interface for you to just open up your images, open up the standard template in either MNI or TaylorX space, overlay the ALE results, and boom and go. So another resource on the BrainMap website, brainmap.org/pubs, is a list of various paradigm-based meta-analysis that have been published in the literature. This is a very, very small and outdated list at this point. These are all meta-analyses that have been published in healthy individuals. There's also a number of meta-analyses in diseases and disorders. And I highly recommend you go check out the list of publications, because if you're interested in publishing an ALE meta-analysis, it's good to make sure that someone hasn't already done one, so that you're taking a unique approach to addressing meta-analysis in the literature. And also, there's a number of really interesting meta-analyses that have been published, and it's nice to have a model to work with while you are working up your own manuscript. So just as a summary, we've been talking a lot in Sleuth of simply working with an idea of a cognitive construct of interest, whether it's working memory or face discrimination, and doing a paradigm-based ALE meta-analysis where you're interested in some task or construct and you go find those studies in the literature and meta-analyze them. And another approach is to take an interest in a particular region of interest. And so later on, Veronica is going to address a ROI-based ALE meta-analysis where you focus on all activations reported in the literature that are within a given region of interest, and that is the meta-analytic connectivity modeling approach, or MACM. An additional resource that's available on the BrainMap database is in relevance to resting state networks. We know that resting state networks are long-range, functionally connected regions that exhibit coherent fluctuations at rest. These are consistently identified using ICA, so I'm sure you're very familiar with ICA resting state networks. The functional interpretations of these networks can be fairly problematic because participants are at rest doing them. But we like to talk a lot about how we can find the sensory motor network or the visual network or the executive control network. BrainMap has done a number of studies where we've utilized the BrainMap database to provide functional labels to these resting state networks. We refer to them as intrinsic connectivity networks because, as demonstrated by Steve Smith in 2009 et al., that they are not limited to just the resting state. So I guess I skipped that. Goodness, this clicker really is frustrating. Okay, so down on the bottom, just a brainmap.org/ICNS. If you are doing resting state studies and you want to try and provide very quantitative functional labels, functional interpretations of those networks, we've provided additional resources to enable you to put a nice functional fingerprint on what those networks are. And so with that, thank you for your time.
Wow, so you've really rushed through it. So we do have actually time for 1 or 2 questions. Any questions from the audience? Yes, please. Wow, I feel bad.
So you can use MNI or TAL, and you have a few algorithms now that you want to switch between.
Correct.
Do you have...
Correct. So if the papers are in the BrainMap database, then we have actually already applied a correction to them so that when you export your coordinates from the Sleuth environment, you have a little preferences option. You can export them in TaylorAct or export them in MNI, and it will all be done automatically. If you want to export into TaylorAct, the ones that were TaylorAct will stay TaylorAct, and the ones that were MNI will be exported in TaylorAct space, vice versa for MNI. If you are doing these things manually, and you want to take a set of coordinates that you have on your own and create a text file for them, say they're in TaylorAct space, if you go into Ginger Ale, then there is a coordinate converter application in the menu that you can load in a text file in TaylorAct space, click a couple buttons, and it outputs it in MNI space so that you have the flexibility of enhancing your Sleuth file manually.
Any additional questions? Then maybe you could briefly comment, because that's one thing I'm getting asked a lot, and I think it may be also relevant to this audience here, the different thresholding options in Ginger Ale. It seems to confuse people at times.
I was anticipating that question, and I was going to gently lob it over to you for the answer. So, yeah, we're not promoting the FDR methodology anymore. We're really pushing the FWE correction, both, and then our lovely Ginger Ale programmer is in the back. Everyone wave at Mick Fox. Woo! And there's nice options for both the voxel-wise and cluster level thresholding.
I think that's a good point. Any other comments on BrainMap and Ginger Ale? If that's not the case, then okay. Last second.
I'm looking through the literature on an investigation I have at the moment, and I see some people put all foci in, positive or negative. Is that to sort of test methodology as opposed to, say, clinical relevance?
Yeah, I mean, I'm a strong advocate for if you're looking at increases and decreases, those need to be addressed in separate meta-analyses. You don't want to conflate effects, because then that just doesn't make sense. You've confounded your meta-analysis. When you combine increases and decreases in the same Ginger Ale, you can just simply keep those separate and run 2 separate meta-analyses.
Okay, thank you.
Maybe as a small add-on to that, and going back to really what Felix said, technically you can do anything.
Absolutely.
But you need to know how you would interpret that then. I think what Angie really mentions is an important point. If you keep them separate, you can interpret them clearly as hypo, let's say patients versus controls.
Right.
Hypo, hyper. Now, you can pool them. Technically, that's no problem. But then you just need to be aware of the fact that this then just means apparent. So you could have one find that's driven by half of the studies that drive it are hyper, the other half is hypo activations. If you are ready to interpret that, then you can do it. But you just need to. I think that can't be stressed enough. And you've done it, and Felix done it. You can do anything technically. You can mix VBM and resting state and fMRI findings. You can just technically pool anything you want. But it always comes down to what does that mean. And as a very, very urgent rule, please think about how you would interpret these things before you run the analysis.
So as an add-on to that, I will say with respect to the increases and decreases, if you look at patients greater than controls and you find the amygdala, and controls greater than patients and you find the amygdala, and then you write a manuscript on it, please address the fact that there is apparent controversy in the literature as to the direction of that effect. Don't just say, hey, the amygdala was found in both, isn't that cool? You know, you need to go in there. You've obviously identified an area that needs to be further researched, and so that's a good opportunity for you to dig a little bit deeper into the publications and try and figure out why you observed the amygdala in both.